So, hi everyone. I'm Jan from Zelezniak Lab at SysBio, which is at Chalmers in Sweden. And I'll be presenting our recent study where we use deep learning to learn the regulatory grammar of DNA and then try to apply these models also for protein expression engineering. So this study was recently accepted in Nature Communications and hopefully will also be published and made available online in the next couple of weeks. So stay tuned for that. So one of the main initial findings in our study is that when we take thousands of RNA sequencing experiments, which cover also the majority of different experimental conditions, and we plot the protein coding genes sorted on the y-axis according to their increasing median gene expression levels, we see that the variation per gene across all of these different conditions and experiments is actually much smaller than the whole dynamic range of expression levels in our data. So this shows that the gene expression is quite well conserved across all the different conditions for each gene, as well as it suggests that this information must somehow be encoded in the DNA sequence itself. So from this gene regulatory structure, as we termed it, we used over 2,000 base pairs of DNA sequence, 64 codon frequencies, and additional mRNA stability variables as input data into the convolutional neural networks. And the target variable were these median gene expression levels, which I described before. And in yeast, our models were able to explain over 82% of the variation of gene expression levels in the test data set. And when we used additional model organisms spanning from bacteria to human, we showed that on average, 60% of the variation of gene expression can be explained or deciphered from the DNA sequence alone. So further on in our paper, we show that this gene regulatory structure is an interacting and co-evolving unit across all of these regions. So next we wondered what our models are actually learning and extracting from the DNA sequences. So we reconstructed the regulatory grammar that they're learning, and we see that it actually spans all of the different regulatory regions and covers all of the known sequence motifs in yeast. And next, we performed association rule analysis. So we treated each gene as a basket of motifs, and we analyzed the co-occurrence of motifs across the genes. And here is an example of 4 genes which share a transcription factor binding site marked blue and carry additional motifs marked red, which co-occur with these transcription factor binding sites. And these red motifs actually repurpose the transcription factor binding site in a range of over 3 orders of magnitude of gene expression levels. So next we wondered if we can somehow use these regulatory models to guide gene expression engineering. And considering the way people usually approach this experimentally is just to take one region, such as a promoter, and swap it out for a well-known strong or weak promoter when they want to fine-tune gene expression, not taking into account any of these other additional regulatory regions. So then we analyzed if we keep the promoters fixed for each gene and just vary these terminator regions, we show that actually just by changing one of these regions, we can unlock a lot of regulatory freedom over 2 orders of magnitude, change of gene expression levels per each gene. So we then tested this experimentally by predicting for a set of native promoters with, we predicted strong and weak terminator variants that can upregulate or downregulate the function of these promoters. So first of all, we see that we have good correlation between our predictions and experimental measurements. And also we show that on average 20% of the gene expression levels can be upregulated or downregulated just by swapping out with these predicted strong or weak terminators. So this shows that we can actually learn the regulatory grammar just from the native genomes and use these models to guide gene expression engineering without the requirement of any synthetic approaches or constructs. So thank you for your attention and interest. And if you want to read more about this study, it's available on bioRxiv, though improved version will hopefully be properly published in a couple of weeks. So stay tuned for that.